Thank you all for coming. My name is Jeff McDonald. I'm the president of the Hydrology section. And on behalf of our president-elect, Scott Tyler, and our secretary, Charlie Luce, and the entire section, I want to welcome you to this set of named lectures. It's a real honor and a pleasure to be able to introduce this event. But before we get started, I wanted to give you a little bit of forewarning about what we're trying to accomplish this afternoon. This is a slightly different format to previous years, and we hope this will be beneficial for the section. We're having the two named lectures, as you can see, and momentarily you'll hear from the first citationist. And then we're transitioning to the hydrology section business meeting and awards celebration. This is co-sponsored with Kawatsi, and this will include featuring other awards at the section level. So please come to the business meeting. It's a ticketed event. I think we've sold out, but for those that have a ticket, we'll greatly look forward to seeing you there. Half of our membership are students. So if you come to the business meeting, make a special point of reaching out to students, making them feel welcome. If you're a student, take it upon yourself to introduce yourself to senior colleagues, and we hope this to be a mingling event. It's an open bar for the first hour, so that should help a lot. And then we will have the business portion of our meeting that will start at 7:30, and I'm hoping this will last 30 minutes. And there'll be then time after that for further refreshments. There will be food. So we hope this will help to build intimacy within our very large section. So without further ado, I want to hand over the stage to Eric Wood, who's going to read the citation for Thorsten Wagener, our Witherspoon lecture this year.
Thank you, Jeff, and I'm really pleased to introduce Professor Thorsten Wagener of Bristol University as the 2017 Paul Witherspoon Lecture. A 2002 civil environmental engineering graduate from Imperial College in London, he did a postdoc at the University of Arizona, was a faculty member at Penn State University, and is currently a professor in the Department of Civil Engineering at Bristol in the UK. Scientifically, is highly cited with major contributions in the following research areas: the analysis and modeling of hydrologic systems, where he characterized the tremendous variability across catchments and quantifying sensitivity, uncertainty, and risk. In the area of diagnostic evaluation of environmental models, and in the integrated assessment and scenario analysis for water resource management. Thereby, his research has spanned the range from hydrologic understanding to model improvement to decision-making. Thorsten has been unselfish in his community service to AGU and the hydrology section by taking leadership roles in the Early Career Hydrology Award Committee, the Horton Research Award Committee, and on union award committees. In leadership in the hydrology community, where he has served on Kawasi's Education and Outreach Advisory Board, for UNESCO committees, for IHS, including the steering group for the project on Ungaged Basins, the PUB initiative. Perhaps very noteworthy was his tireless contributions to the PUB decade, culminating in his co-authorship of the 2013 book, Predictions in Ungaged Basins, a synthesis report across processes, places, and scales. That was published a few years ago by Cambridge University Press. His work has already had a profound influence on the evolution of our field. It's clear that his work will have a continued impact. Let us welcome Thorsten Wagener as the 2017 Paul Witherspoon Lecture.
Thank you very much, Eric, for those nice words. When I was told about the Paul Witherspoon Award for mid-career scientists, I had to think about it for a moment and think about what am I a mid-career scientist and what is a mid-career scientist? But I realized that I'm pretty much exactly 20 years after starting my PhD now, and I'm 20 years away from retirement. So I'm really pretty much a mid-career scientist, even though the announcement of possible changes to the UK pension scheme for academics might mean that I'm back to being an early career scientist because I might have to work a few years longer, it seems. I'd like to start by thanking a few people. First of all, my mentors. So from my undergrad in Germany, Jürgen Jensen and Gerd Försch, who introduced me to the area of hydrology and got me interested in water as a research topic. My mentors at the TU Delft, Peter van der Veer and Tom Rientjes, who then got me interested in using computers. And then ultimately my PhD advisor, Howard Wieter, and my postdoc advisor, Hoshin Gupta, who then helped me understand how I combine the two interests to actually do research that hopefully is meaningful. I thought about who I have to thank. I'd like to thank Eric and as well as Richard Vogel and Keith Bevan for nominating me for this award. And I started making a long list that is certainly not complete of people who either helped me with constructive criticism or worked with me in different projects or papers. Sort of I started with some of the people more older than me, and then I continued with the people who are sort of my peers at my level. And I think one of the things that I was particularly lucky with is that at my PhD graduation time period, there is really a great number of really good hydrologists, and we sort of grew up together over the last 15 years, and a lot of them have become very good friends. And then finally, of course, I started collecting all the students and postdocs that I had over the years, and whose work I mainly have been presenting over the last decade or so, and I will also continue presenting their work tonight. So today, for this lecture, I would like to talk about making a bridge, or really two bridges. One is from catchment scale hydrology, or small scale hydrology, I should say, to numerical models at much larger domain scale, and from hydrologic processes to the impact of climate on correlated biological process, in this case, infectious diseases, waterborne infectious diseases. As a Paul Witherspoon lecturer, I would like to start this by saying a few words about Paul Witherspoon, who I have not met in the past. I never had the fortune, but he was active at UC Berkeley and Lawrence Berkeley National Laboratory, and through reading about him, for watching this great time capsule set of interviews with famous hydrogeologists that IAH has put up, I've learned that the first group of students he had was Alan Fries, Shlomo Newman, and Jirai Javandel. It seems like your career peaks early if you have that kind of PhD students as a starting assistant professor. What particularly fascinated me in the interviews I listened to was his early recognition for computers, the power of computers and mathematics in hydrology, in understanding hydrology and using it. And I particularly liked how he integrated learning into his research, into his teaching. So he sent off his students to the maths departments to take courses in more advanced mathematics than he had ever taken, so that they could come back and teach him the mathematics he needed. And he brought the same kind of reverse classroom approach into his classroom teaching, where he regularly had the students teach to him rather than the other way around, something we're trying to do today, but he's done it a long time ago. His obituary in EOS by Alan Fries, Shlomo Newman, and Jirai Javandel had this beautiful sentence in it: to enter Paul's orbit was to experience a stimulating mix of high intelligence, deep curiosity, and love of life.
to talk.
Okay.
There you go.
Wave in the back if you can't hear me. So following from some initial paper that Alan Fries wrote with Paul Witherspoon on regional groundwater modeling, just a year later, Fries and Haaland, of course, published their 1996 Journal of Hydrology blueprint for physically based modeling, in which they discuss, among other things, the purpose of hydrologic models. And they made this list here to synthesize past hydrologic events, to predict future hydrologic events, to evaluate effects of artificial changes to the hydrologic regime, and to provide a means of research for improving our understanding of hydrology in general. Now, while these might not have changed that much, I think that certainly the emerging water management challenges that we have now push us from the catchment scale to much larger scale continental and global domains. So the questions we need to ask then is, what does this mean for how we build and test such models? How do we move from catchment scale hydrology to scales of continents? What particularly interests me is how models achieve good performance, which has long been considered relevant for hypothesis testing. So the third and fourth part, if you like, the third and fourth task that Fries and Haaland set out. So this is a sentence from James Kirchner's paper. Advancing the science of hydrology will require not only developing theories that get the right answers, but also testing whether they get the right answers for the right reasons. And of course, that's building on work by others like Clemish. And I think that's key to what I want to talk about today. The way I approached this in the past was to look at consistency between the model and the underlying system as something that is more important than optimality with some objective function that might not be particularly robust. I think while we might in the past have looked at how models achieve their good performance or how models reproduce the system, might be mainly focused on hypothesis testing, but I think given the growing challenges like climate change and other issues that focus us to look at a changing world, push us to look at hydrologic models and how they function as a central question when we go forward in model testing and building. So the way we have done this in the past is looking at sensitivity analysis tools, sort of mathematical techniques that look at how the output of a model can be attributed to variations of its input. And the example we could think of is that we might have different algorithm choices, different choices of how we represent a particular process. And the question is whether these choices matter given uncertainty in parameters or input forcing. So as a visualization of this idea, so in addition to asking how well does the model perform, we ask how does the model achieve this performance. I like to use this fish. It's a reef triggerfish, and I'm not going to pronounce the Hawaiian name that it has. Maybe somebody can tell me afterwards how it is actually pronounced. I don't know. But this fish has very interesting behavior. So if it sees a predator approaching, then what it does is it will find an area between rocks, crevices within the rocks, and it will use its fins to lock itself into the rock in a very rigid way that the fish, the predator, even if it grabs him at the tail, cannot actually pull him out. So we can observe the behavior of the fish as much as we can observe the input-output behavior of the model. But really, what we really want to know is how does the fish capable of doing this? So if we have X-ray, if we can look inside the fish, then we can see that it has two fins at the top, that if these two fins connect, then it creates a very rigid structure of its skeleton inside, which allows him to have this lock himself into the rock. So the question that I therefore want to ask for the rest of my talk is who's in control? Now, I'm asking myself that question a lot these days, but I will stick to hydrology for the rest of my talk. And the demonstration or the examples that I use are two. So both of these are projects that we've worked on for several years, and I would like to talk a little bit about the evolution of how we got from beginning to end. The first part is about dominant process controls as a key to minimally parameterized models. And so this is about building models at the larger scale, parsimonious models at the larger scale. And the second part is modeling process controls as key to robust impact assessment. And if you're wondering why there is a sheep, I will tell you in a little bit. The first project started, or the first part, the research for this first part started a few years ago when I had the great pleasure that Markus Weiler of the University of Freiburg asked me to be co-supervisor of a PhD student of his, Andreas Hartmann, and that initial research evolved to a long-term collaboration through his postdoc at the University of Bristol, and now he's back in Freiburg and runs his own Epkarst research group. The second person who contributed to this research mainly is Fanny Sarasin, a joint PhD student at the University of Bristol. So what we looked at here is how do we model karst hydrology? So chemical weather of carbonate rock and other physical processes develops preferential pathways that are shown here. And unfortunately, I can only point to one side. So we see these at the surface, and then we see a mix of diffuse flow pathways and concentrated flow pathways in the subsurface through this process. I'll give a couple of examples in the beginning of this whole system, but mainly I want to talk about the upper part, the upper layer, which is the Epkarst. This Epkarst develops due to the dissolution activity of the carbonate rock near the surface, and it produces these kind of soil pockets that you see here on the left and the Epkarst, and you see on the right that it creates a mixture of diffuse recharge from the surface and concentrated recharge from the surface. This Epkarst controls recharge processes with significant subsurface heterogeneity, including lateral flow concentration to these concentrated recharge points. Karst areas represent an important source of water for the supply of many countries. So on the left you see a scatter plot which shows carbonate rock outcrops versus karst water percentage of total water supply for a range of countries. And you see that quite a range of countries, particularly Eastern Europe, has a significant amount of water from these karst regions. And in some parts of the world, these kind of karst springs are already running dry. So this is an example of fire spring from Palestine, where excessive pumping has reduced the outflow from the spring so much that it has completely dried up. So the question that we went after for quite a few years is how can we use what we learned about complex and heterogeneous systems such as these karst systems at the small scale. I call it catchment scale. I know it's not really a topographic catchment certainly, but it's as a reflection of the small scale, to build minimally parameterized models for much larger domains. So we want to go from this kind of image of a spring towards this domain ultimately, which includes Europe, North Africa, and some of the Middle East. So the starting point in this process was looking at some of the data sets that are particularly rich in experimental locations. In this case, somewhere in Israel, a karstic cave at Mount Carmel, which has a unique set of spatially distributed flow and tracer observations, these kind of trip observations into the cave, so that it's a fantastic opportunity to look at recharge patterns and variability of recharge in space and time. At that time, early on in his PhD, Andreas and Markus Weiler developed the idea of a very parsimonious, so minimally parameterized and flexible model structure based on distribution functions that they were able to use to reproduce the behavior to fit the data. Now that gives us a model at one particular place where we have lots of data. So the next step then was to find other locations, in this case in Austria, Switzerland, and around the Mediterranean, to see whether this relatively simple model structure was capable of reproducing the behavior of a wider range of systems. So in this case, different experimental sites which had hydrological and hydrochemical observations that could be used to derive signatures from, and that we can see, compare the model with. So that gives us an idea of how well the model performs. But in addition to that, we wanted to also be sure that the model had only parameters that were relevant for reproducing the behavior of the system at least some of the time, so that the model was actually parameterized efficiently while being sufficiently flexible to represent a wider range of systems. So what you see here are for 3 of the locations. On the vertical axis, you see the signatures, so hydrological and hydrochemical characteristics of the outflow. And on the horizontal axis, you see the different parameters of the model. And the colors indicate, in this case, a global sensitivity analysis index that I'm not going to get into, but essentially the more red, the more varying the parameter has an impact on the output of the model. So what we wanted to see is whether each of the parameters here has above it at least sometimes a red field, indicating that it's relevant in reproducing some of the behavior and therefore needed in reproducing the system. You also see that there's quite some variability between the different systems that reflects that their behavior is quite different. And the other thing is, of course, to understand how these different controls vary across the system. So that gets us from one place to many places, or a range of places, but all these places still are very data-rich, if you like. Ultimately, what we wanted to do is we wanted to build a model for this, all the blue area you see on this map, which are carbonate rock outcrops in our domain. So we moved from a model that represents a relatively well-observed system to applying the same kind of idea at a grid scale across this domain. The problem, of course, or one of the issues with that is that now, of course, we are lacking the kind of rich hydrological and hydrochemical data that we can use to observe, to calibrate the model. So the approach we took to compensate for that is to start by sampling from a wide range of parameters, so including the uncertainty in the parameters that we had, and then developing essentially a system of different perceptual models that reflected the variability in a very crude way of karst systems that we would expect in our domain. So in the top left, we have the blue are humid hills and plains, so with relatively thick soils and not particularly strong gradients. Below that, Mediterranean medium-range mountains, and on the right, the green high-range mountains and desert hill and plains with very shallow soils. So the idea was that we could then use hydrologic similarity principles to essentially regionalize information from FluxNet and other FluxNet sites to these places as soft rules. So the idea is to simply make sure that the model reproduces the same kind of, let's say, seasonality in the soil moisture and seasonality in the actual evapotranspiration in a very limited way. So relatively weak constraints on the model. That reduced the uncertainty considerably, and on top of that, we then applied for some of the parameters that we had measurements for, or that were linked to physical characteristics that are observable. We then constrained them subsequently with that information to make sure that the dynamic constraints and the physical constraints were consistent. The resulting model across the domain we then tested further. Of course, they're not really observations we can directly use, but in this case, we collected from the literature recharge estimates in different places across the domain that either were derived with other kinds of models or through observations to compare our models against even just at an annual timescale. And what you see here are the two different models on the right, the heterogeneous model with the subsurface heterogeneity of diffuse recharge and concentrated recharge and lateral flow concentration, and below that the more typically applied large-scale model with homogeneous subsurface characteristics. And if we compare what I call the observed data here and the simulated data, then you see that the yellow line at the bottom would be a homogeneous model, so it would not produce sufficient recharge. The blue is the heterogeneous model. And then if you switch off the heterogeneity, so I should say that the homogeneous model that we used initially was PCR Globe, so one of the widely used global hydrologic models. And if we switch off the heterogeneity from our model so that this becomes a homogeneous recharge behavior, then we see it becomes very close to the other homogeneous representation, suggesting that this kind of process representation is relevant for the model. If we then drive the homogeneous and the heterogeneous model with climate projections, then we see that they provide significant differences. For one, the estimates of recharge of the heterogeneous model are higher. The variability is larger, so the error bands or the bands of variability that you see, the gray areas, are much larger. And also the response to changing climate is a bit different between the two models. So where we're now going with this is making other kind of hypotheses at these large scales and making more processes explicit. In this case, vegetation, where we're adding explicit to the vegetation to the model that before had an implicit ET scheme, so that we can actually look at land use change versus climate change impacts for these particular regions of the world. So the process we went through in this research was an attempt to learn a range of things. I tried to generalize 3 of them in particular that interested me, and the references underneath it are mainly other papers or people that try to do something similar but in different ways that I find are particularly interesting to compare against. So the first thing we're trying to achieve was to develop and test hypotheses about controls at large scales. So using what we've learned at small scales and transferring this to a larger scale, making hypotheses there that we maybe can compare to some proxy data, or if that's not possible, at least see whether including these hypotheses makes a significant difference. The second part is to advance evaluation strategies that enable us to better understand how a model reproduces the results rather than how well it does so. I think that's a really important area for advancement, particularly if we go to even larger domain models that put a very high computational burden on understanding and analyze these models in detail. But so the more richer the model gets in what we could learn from it, the less we're able to understand it and extract it from the model at the moment. And the third part is to develop minimally parameterized and flexible process descriptions that are suitable for the large scale. So if we have minimally parameterized models, we have a higher chance of understanding where these models fail and where they work, and we have a much better chance of finding sufficient information maybe to constrain the parameters. So I'd like to refer to one talk of one of my colleagues, Raphael Rosolem, on Friday, who will talk about work we're currently doing with Shams Rahman and Stefan Collett on developing groundwater schemes that are more efficient and can be used maybe in Earth system models at large scales. So moving to the second part and to the image of the sheep. So here the idea is to look at process controls as a key to robust impact assessment of climate change. And the sheep is there because what we're looking at is waterborne infectious diseases, particularly in relation to livestock, to sheep and cows. And I'd like to refer to another talk, but that's given by my PhD student Ludovica Beltrame on this topic. Unfortunately, it's in the middle of Bob Hirsch's talk, Langbein lecture, so you will miss it, I'm afraid. So the starting point here was one of the starting points for me was a very short editorial that Eric Barron wrote some years ago. And in that editorial in Science, he asked, how can we provide the environmental intelligence to support decision-making in relation to climate change? How can we provide information at the appropriate temporal and spatial scales for local and regional decision-making? And the area where we're interested in seeing how we can provide this environmental intelligence is waterborne infectious diseases. A number of recent review papers in the area of infectious diseases identify three main challenges for that area at the moment, particularly in the context of assessing climate change impacts. The first one was, how do we disentangle the effect of multiple non-biological drivers of infectious disease risk? So things like climate, socioeconomic characteristics, land use, soil pH. The second is, how do we describe the mechanisms linking drivers and disease transmission? Because as you will see in a moment, the largest chunk of models currently used is based on empirical correlations. And the third question is, how do we then identify the relevant space and time scale so that we can actually enable and use these models for management and to understand the intervention potential in the context of infectious diseases. So the particular infectious disease that I would like to focus on is called fasciolosis. It's caused by a parasitic flatworm, Fasciola hepatica, also called the common liver fluke, which is this thing here. It's not quite as large as it appears here. It's only 3 centimeters long. And it attaches itself to the livers of sheep and cow. What I've shown here is the life cycle of this parasite. So if you have an affected animal that releases eggs onto the pasture, then if these get enough moisture and the temperature is right, then these eggs will hatch. It will release Miracidia, who will actively search a host. That host in this case is a mud snail, and the Miracidia will develop in the snail and then eventually leave the snail as Metacicaria that then survive on the pasture and get ingested by other animals and therefore infect the next cow or sheep. Fasciolosis is spread around the world, infecting livestock, and it's even emerging as a zoonosis, as a disease that also infects humans, particularly in developing countries. It costs the livestock industry globally about $3 billion. There's no vaccine for this disease, and it shows increasing resistance for existing drugs, as many other diseases that we're aware of. And it's likely to change significantly in the context of climate change. On the right, you can see in green the distribution of fasciolosis, and you see it's spread pretty much everywhere. So the current type of models that is used to look at risk also in the context of climate change are these kind of empirical relationships like the Olorenshaw index, which relates monthly climate variables, rain and temperature, to a fluke risk index based on observations from an earlier period in the 50s and 60s. And the current practice is to use these kind of models to then drive them with climate change projection to look forward and see how the risk might change. So what you see here is an example from the UK, where you see summer and winter images that show for this particular study at 25 kilometer resolution how the risk is growing in certain parts of the UK for infection from this disease. Now, the problem with this kind of empirical model is, of course, that one, it's difficult or questionable whether we should use it to extrapolate far beyond the period for which we have observations. It doesn't allow us to look at interventions. For example, if there is some kind of treatment that we can do, what impact that might have. And it also does not allow us to look at the right scale, because it will be at a very large scale, and I will say why that is a problem in a moment. Beyond fasciolosis that I'm looking at, there were recent reviews of other how waterborne diseases are approached in general, and this is a summary. Here you see in this table a review of 15 years of papers in this area and the kind of variables that are used to predict infection risk. And what you see is, of course, that at the bottom, the vast majority of these papers uses rainfall and temperature, and then a whole other range occasionally for predicting risk of infection. If you look overall, then the distribution between empirical and mechanistic approaches is biased, strongly biased to empirical approaches, and a lot of the mechanistic approaches are more very recent strategies. So one that I want to point out, of course, Andrea Rinaldo and his group have done a lot of work in this area on mechanistic modeling of waterborne diseases, particularly in the context of controls exerted by the river network or waterways. And there's just a new review paper on this topic just published in Advancing Water Resources. So the way we approached this problem was, rather than in an empirical manner, trying to build a mechanistic model. So what we did is, in a very simple, shortened way as explanation, we coupled a distributed hydrologic model, a top model version that's driven by daily temperature and rainfall, with a life cycle model of liver fluke. The reason why we use liver fluke is because it's one of the more well-studied parasites. The connection between the two is soil moisture, and the model ultimately produces the number of infected metacercaria on pasture that could be ingested by livestock. I'd like to give you two examples of how this model works differently from the previous approaches. So this is a change in temporal response of the model. This is for the River Tau in Wales, and what you see here is a time series of, in light gray, mean winter temperature, and in dark gray, the number of winter days above 10 degrees Celsius. And 10 degrees Celsius is one of the thresholds for both the snail and for the metacercaria to be more active. And if we compare these two years to the year 2002 and 2007, between the empirical model, the All-in-Show model, and our mechanistic model, then how do these models respond differently? The 2002 and 2007, you see that the mean temperature is roughly the same, but in 2007, the number of winter days above 10 degrees is much higher. If we then compare the response of the two models, so we have in black the All-in-Show index, that goes up to 100, and in blue our model that gives you actually the number of metacercaria. Then you see that for the 2002 year, in the beginning of the season, our model produces a very low count when the risk from the All-in-Show index is still at 100%. But then in the subsequent year, you see that, or in 2007, you see that while the Olenshaw index is about the same. The mechanistic model shows already twice the number of infected metastacaria to be present. So what that means is that it's a tool to enable us to look at how increasing variability might impact infectious disease. The idea, well, the reason why this is relevant is, for example, for the UK, the projections are that the variability will grow faster in climate, will grow faster than the mean values. The other aspect is then next to the temporal variability is the spatial variability. So on the left is an example of the Olenshaw index applied with typical weather data that we might have in the UK for the same catchment. And what you see is that you see variability here of risk from light gray to red that varies with the climate, or in this case rainfall gradient, across the catchment. Now the problem is if you had two catchments with the same climate but completely different topography, for example, you would get exactly the same kind of risk estimate. That means if you wanted to use the Olenshaw model in these two catchments, you need subjective judgments to say that essentially the same amount of risk would mean something more for one catchment than for the other. So there is, the proxy is quite removed from what you actually want to know. In case of the mechanistic model, we can run this at a much higher resolution, in this case 25 meters. And what you see is the same kind of variability. It's not so, doesn't come out as well here, but essentially the increasing climate driver you can see, or weather driver you can see here as well. But what you see is the spatial variability impact of topography on which parts of the catchment are actually likely to be at risk and which ones are not. What we can do now is we can aggregate the cells of this model to the farm scale so that we can look at farm level risk, which we cannot do with the previous model. So some conclusions on this part, or some challenges and opportunities, if you like. I think that there's a lot of opportunity in this case we used infectious diseases, but other areas of maybe climate change or other change impact that relate or are driven by hydrologic processes to maybe not completely build a mechanistic model, but bring the hydrologic drivers much closer to what is actually driving the system than what it is at the moment. So in this case, we went from monthly temperature and rainfall to soil moisture as the driver. Now, there are lots of challenges. Data availability is certainly poor when you look at infectious diseases, and the value of existing data is difficult to assess. It gives you, working with infectious disease data gives you a new appreciation for hydrologic data. Different communities have very different approaches to model evaluation and model validation, so that doesn't necessarily translate very well. What we do with hydrologic model is not the same what people do in the epidemiology. And it's also a very time-consuming process because it means learning a range of languages. As you can see here, you have to learn biologist, hydrologist, vet, infectious disease model, and probably some other things in order to communicate between these groups. So in closing, I think a focus on how our models predict in addition to how well our models predict and how consistent these simulations are, these simulated controls are with our perception of reality offers a lot of interesting research opportunities. The late biologist Stephen Jay Gould said that diversity is nature's principal theme. And while I do not think that there's a silver bullet out there for hydrologists to understand this diversity and what it means for hydrology, what I mean is maybe not the one model or the one observation or maybe the one theory to explain all of that diversity to us. But I think there is an opportunity for a process of maybe painstakingly mapping and organizing this diversity for us, the spatial and temporal variability of controlling processes as an advancement in hydrology overall. Thank you very much.
Well, thank you very much, Thorsten. I think in hydrology we've avoided karst systems like the plague. And what you've heard today is an embracing of both karst systems, and maybe not the plague, but at least infectious disease. So, Thorsten, thanks so much. We do have a little time for questions. It's a little awkward because we're filming, we're live streaming, we're recording, and I know the lights aren't completely conducive. I'm hoping we can turn the house lights up if that's possible. There's a microphone down here in the center. I'm not sure if others are scattered around, but the spotlight's shining on the stage here. But I'd like to chair a discussion and hear from questions that the audience might have. And I'm going to look for hands, and if you're interested, maybe make your way to the mic and kind of position yourself near there to ask a question or two of Thorsten. And maybe while people are doing that, you're going to help. Okay, great. I'll just maybe kick off, Thorsten, as we wait for people to make their way to the microphone. One of the things that's been discussed a lot this week is how to bring something like lateral flow into a CLM or some landscape model. And this is really much like your first half of the talk, where you're working with karst systems that are extremely difficult in ungauged basins. How do we bring something like complex process into a model that is, we want it to be minimally parameterized, like you were discussing? So that seems to be such a grand challenge, even beyond karst systems, as it relates to some of these community questions. Any thoughts on that?
Yeah, thanks. Easy. Well, I think the approach that we took in the work so far was by not shying away of making hypotheses, and trying to actually test whether it can work. So I think we're sticking very closely, very often to the system where we have lots of observations because we can test them. But I think there is an opportunity in addition to looking at highly observed systems, to simply dare and go out there and think with, particularly with the experience of process hydrologists or experimental hydrologists like yourself, and simply dare to make a guess at what is happening at the large scale. And I think what we can add are methods and ways to analyze whether that makes a difference and under what circumstances it makes a difference.
All right, thank you. Other questions?
I would say, unfortunately, our microphone is not working, so if you do have a question, just raise your hand and stand up and shout loudly. So,
Charlie?
Sure, and unfortunately it's going to come across a little too close to Jeff's. But I was looking through the results you were getting from the disease model, and did you learn enough from the very fine scale model to be able to come up with a formulation that can be applicable to places where they can't afford to do the top modeling and the detailed work? Is there something that can be done at that broader? Did you learn enough to scale that up?
Well, I think in terms of the mechanistic model, the thing is the hydrology is not so complicated to do. I think that was one of the main interesting aspects of that, and one of the main opportunities is that we can use a relatively standard hydrologic model to do something we think we can do reasonably well, and at least in places that are humid and have the right processes with this kind of modeling strategy. So I think we're not limited there. I think we need more, always can use more and better data to understand whether the soil moisture patterns are right and things like that. But I think given the uncertainty in some of the data on infectious diseases, I'm not sure how much that will actually help. I think overall it won't.
Okay, thanks. Again, I'm scanning the crowd, looking for hands. Yell out if you have a question. Yes, Ross.
Has the modeling of the infectious diseases part, is it now ahead of the ability to collect data to govern that?
repeat the question. Repeat the
Yeah,
question.
the question was whether the model, the infectious disease model, is ahead of the ability to collect data. I think yes is the simple answer. I think so. I think the problem is that there isn't necessarily a synthesis of all the data that is there that is easily accessible, which I think would be the much better next step to compare against. So we have a set of data that we use, which I didn't show, to compare the model against, but I think that going forward, integrating existing data would be very good, particularly because getting data on some of the aspects of the disease in the field is very time-consuming, let's say.
Thanks, Thorsten. Another question? Yeah. Scott?
Yeah. Thank you, Thorsten. I appreciate the talk. I have a question kind of on the epidemiological results and a little bit on sort of questions of modeling. When you showed the two results, the empirical model and your model, someone was biased in those results in time. I don't know who. Someone was ahead or someone was behind. And the question is, an empirical model is generally designed not to be biased in time. So who's right?
Well, the time difference, which I didn't explain, is there because the models don't predict the same thing exactly. So the Euler-Shaw model predicts whether the conditions are right, let's say, in terms of the climate to produce metastasia, but there will be a time delay before that actually occurs, and that's why the mechanistic model goes up later, because it includes that time delay of response in the biology to actually be infectious, if you like. So that's where the delay comes from.
Good. Okay. All right. Well, I think we'll end there and thank Thorsten again for a really informative lecture. We'll be presenting plaques following the two lectures. So if you're eager to speak to both speakers, just give us 5 or 10 minutes to click a picture or two. I want to give you a sense of the agenda. I know we have a lot of people in the room that perhaps weren't here at the beginning of the last hour. You see the agenda for the afternoon's activities. We're about to start the Langbine lecture. You'll be hearing from the citationist, Dennis Lettenmeyer, momentarily. But I want to also draw your attention to the hydrology section business meeting, and this will focus on award celebration for our section awards, the Hydrological Sciences Award, Early Career Award, and the Horton Research Grants. Just to remind you, it's an open bar for the first hour, which should be hopefully a great attractor. It's also co-sponsored with Kawazi this year, and there will be appetizers and other food items there for you. So again, we hope you'll come and enjoy that. We hope there will be good mingling and interaction, particularly with our student cohort, and look forward to seeing you there. So I think we're going to just give ourselves a few moments before we start. Again, we're live streamed and taping, and there's some changeover happening now. So we're going to take just a 3-minute break so that we can start right at 5 o'clock. So I'll be coming back in 3 minutes to chat with you.